Hello everybody, welcome back to Learning Biology 14.2. In this relatively short section, we're going to learn about measurement. Alright, so now you may have a plant and you said that it has grown. But how do I know that? This would require us to carry out measurements. And what are we measuring? Usually we need to measure their parameters. So what exactly is parameter? 一個參數啦。So parameter is just like a reference number, 一個參考嘅數字. So therefore, after reading the parameter, after knowing the result, you will know that, oh, this plant or this organism has actually grown or not. Alright? But of course, measurement will not be only carried out one time. One time is not enough. So sometimes we may need to measure it several times over a period of time. Okay? But then, of course, you will measuring it like this second, and then next 5 seconds, measure again, next 5 seconds, measure again, because there will be no differences likely, right? So therefore, we also need a suitable intervals, 一個間距啦, between different measurements. Okay, so the first parameter we're going to use is about size. So when we talk about size, it is not talking about the size of the clothes, S, M, L, things like that. Alright? Instead, below this word size, there will be 3 different parameters. And somehow these 3 parameters are related. I'll give you a hint and you'll try to tell me what will be their relationship in class. Alright? So the first one is about length and height. The second one will be the surface area. The last one will be the volume. So for the hint, you can think about their units. Alright? So length and height is very easy. It's what you know. So we may measure the height of a human, the length of certain structures, like the roots, like the stems of plants. So surface area, usually we won't measure humans' surface area because, for example, you won't collect all the skin of a certain human and try to measure the surface area of that person, right? I assume. I don't know what is the importance of doing this, except that you have a special interest, alright? But if you have that special interest, don't let me know, okay? So usually we measure the flat structures, like the leaf. For volume, this time we can measure the volume of a human, alright? And usually because we have irregular shapes, okay? So that's all for the size. But then, since size is a very usual thing we use, common thing we use, right? So there will be its advantages and disadvantages, alright? So this part, 14.2, is very important to know different parameters, advantages and disadvantages. But then you'll find that some of them are actually like repeating again and again when you see the other parameters. Okay, so the advantages. First of all, like after I measure your height, after I throw you into the water and measure your volume, will you be dead? No, right? So you remain alive, okay? And therefore, since the organism is alive, the same organism, like you, I can measure you continuously. And you have actually done that since your primary school. Always you will measure your height, like year after year, right? And besides, why is that? Because this measurement is very easy and convenient. Alright? And then for disadvantages, this is because, as you can see, there is a relationship between them. And somehow, I could only get one dimension. I could only measure one dimension of that organism. So that means not all the dimension can be taken into account. Taking into account means can be considered. Alright? Shrub we're talking about here. So a shrub is just like a bush, 一個草叢仔啦. Okay? So it may grow to the left and to the right. So it is growing laterally and without increasing in height. Therefore, like if we are measuring the height of this bush, then we may think that this shrub has not grown at all. Alright, but that is wrong as well. Therefore, we need to be careful when we choose using this parameter to measure. Alright, but then you may have this question. You may ask like, why don't we measure the width or how laterally grown the plant is? Alright, well that is because before you measure, you may not know which parameter is the best. All right, you may not know whether you should measure the height or whether you should measure the width. All right, so therefore you need to like choose carefully. All right, that's all about the first parameter about the size. Okay, and then we move on to the next one that is about the fresh mass. Okay, so when we talk about the fresh mass, first of all, the definition. What does it mean? Of course, it is the mass. That means the weight of the organism. All right, but this time we include 包括咗啦 the water content. Okay, all the water inside. So it's very easy to understand. So how do we measure our fresh mass? You just step on the balance. Okay, 喺個磅上邊啦 and you will know your fresh mass. All right. Okay, so what are the advantages? You can see that they're very much similar. So the organism is, of course, alive, and you can measure this continuously. Okay, and this method is very easy and convenient. You can think back like, okay, so back when you are like in the primary school, when you go for the body checks, and you would always tell your mom and dad not to eat lunch before your body checkup. All right, you would eat like a tea or you have lunch with them after the body checkup. Because in that case, when you step on the balance, you will have a smaller weight or you will have a small number. All right, 你會輕啲啦. And however, what would the disadvantage be? Okay, so this is related to the example. Why did you tell your mom and dad not to have a meal before you go to have the body checkup? Or you may even try to go to the washroom, okay, go to the toilet before your body checkup. That is because it is easily affected by the water content. Easily affected. So for example, like if we're talking about plants, the time you measure the fresh mass of the plant, like before you give water to it, or after you give water to it, both are like the fresh mass, right? But then the fresh mass you get will be very, very different. Okay, so this is the first one. So how is the water content different? It will be affected by like the temperature, okay, and the availability of water. Just like you water it 10 days ago, you water it just now. Okay, so that will be the difference. And the same goes for the animals, just like us. So our fresh mass will be affected by feeding. So whether it is after a meal or before a meal, okay, or excretion. Before you go to the washroom, after you go to the washroom. Okay, so that's all about the fresh mass. And last part, we're going to talk about the dry mass. You can guess that this time we're still measuring the mass, but then this time all the water is removed. So what does that mean? That means we're only measuring the mass of the organic matter. Okay, so you need to know this one about the mass of the organic matter, or we can use another word called the biomass. So in case you want to know what does it mean by organic, that means C linking up with a H, carbon and hydrogen. They're linked up, they're joined together by a certain bond. Okay, 有個聯繫啦. So how do we get the dry mass, the next part? As you can see, the following diagrams, it is quite complicated. It is not difficult to carry out, but it takes a lot of time. So what is happening? First of all, we need to clean it. You need to rinse, make sure that other than the organism, and there will be nothing else. Okay? And then you heat it at around 100 degrees Celsius. So you can think about, why is it 100 degrees Celsius? Why is it not like 1000 degrees Celsius? Because we only want to remove the water inside the organism. Okay? So we are not like burning it. Alright? So, and then after we like put it into the oven for the first time, we put it into the plastic bag so that it will not absorb water from the atmosphere again. Okay, so let it cool. And then we measure the weight. However, it's not like you finish this with one cycle. You need to keep on like putting it into the oven, letting it cool, measuring the weight, putting it back into the oven, letting it cool, measuring the weight again and again until when? Until you get a constant mass. Constant, that means it is the same. Okay, you reach a constant mass. Alright, so as you can see, this method is commonly used for microorganisms. So they are very small in size. Seeds and seedlings, also quite small in size. Okay, so next part, also the last part, we're going to talk about the advantage and disadvantages. First of all, as you can see, the process to prepare the dry mass is very complicated. Okay, so therefore, it is a very accurate method to measure the organic matter. Okay, and then we move on to talk about the disadvantage. Because after you heat it like 1 time, 2 times, as long as there is no more water in the organism, it is dead. Okay, so it is killed by heating or dehydration. That means you take out all of the water. Therefore, since you have killed the organism, you cannot measure it like for several times. You can only measure it for 1 time. So therefore, you cannot carry out this continuous measurement. And then the next one, since like one species, one organism can only give you 1 sample. Therefore, every time you want to measure the dry mass, you need to get a large sample size. Okay, because like in every experiment, you cannot just give me the data of 1 sample size. That sample size is too small. Therefore, not valid, or we say not convincing. 唔夠說服力嘅. Okay, we always need a larger sample size. And last but not least, as you may know, when you look at the procedures, they are very complicated, tedious, alright? So it's not okay for large organisms, or it would take like forever to get the dry mass of 1 large organism. And it is also a very time-consuming, 好耗時嘅一個 method 啦. Alright? So that's all about the different parameters. So we are talking about the 3 parameters, the size, the fresh mass, and dry mass. Alright, so that's all about this 14.2. Please finish these questions, and if you have any question, please feel free to ask during lesson. That's all, and I'll see you again during class. Bye bye.